Hi, everyone, and thanks for joining us. I'm Leslie Bebko, webinar host and moderator for Life Science Connect. Over the next hour, we're going to be talking about delivering process robustness and scalability in single-use bioreactors. We've brought in Qingwei Luo, the Process Development Manager, and Kevin Mullen, the Product Director for DynaDrive S.U.B.s, to explain the process robustness and vessel capabilities role in a seamless system transfer. We're going to talk about sub-process comparability and give examples of processes developed from XDR and HyPerforma S.U.B.s that were successfully handed down to DynaDrive S.U.B.s. We're going to be holding a live Q&A session following this presentation, so please submit your questions as we go along. We'll get to as many of them as possible, and any that we can't get to, we'll follow up with afterwards via email. If you run into any technical issues, please try refreshing your browser first, and if you need additional help, let me know via the Q&A. With that being said, I'm going to turn it over to Kevin to get us started.
Great, thank you. Hello, everyone. My name is Kevin Mullen. As mentioned, I'm the director of the DynaDrive product line, and I'm based in Logan, Utah. Before we get into the data that Qingwei will present, I wanted to give you a little bit of background on the DynaDrive Single-Use Bioreactor. The DynaDrive is a new reactor that we launched about 18 months ago. We still support our HyPerforma single-use bioreactor and will continue to do so. But we launched the DynaDrive bioreactor as our next generation single-use bioreactor, mainly because we needed to increase the scale of single-use and also increase the capability within single-use bioreactors. And so hopefully a lot of you have seen some of our literature on DynaDrive already. If not, I'll take a few slides here and introduce it to you. Just so you know, the DynaDrive has 4 sizes: a 50-liter, a 500-liter, a 3,000-liter, and a 5,000-liter. We've seen really good cell growth in all of these sizes, and Qingwei will cover the scalability from 50 to 5,000 liter on different CHO cell lines that we used in the DynaDrive and our legacy high-performance single-use bioreactors as well as AcceleraX bioreactors. Not only does the DynaDrive have a great scale up to 3,000 liters and 5,000 liters, we were able to enable better turndown ratios within the reactors themselves. The 50-liter has a 10-to-1 turndown ratio, and the larger sizes have a 20-to-1 turndown ratio. That has helped us speed up the process within your seed train from your working cell bank to the reactor. Also, within the single-use reactors, we've increased the power and the mix and the KLA, the mass transfer capabilities within the reactor. We've more than doubled those. And so these DynaDrive reactors are really state-of-the-art, next-generation single-use bioreactors that bring capability of high cell densities with low shear and better seed train capabilities to your process, and I'm excited to introduce more of that to you today. We really took a lot of time in designing the reactors to make them user-friendly. Pictured here is our 50-liter with some of the highlights of the design. Now, some of the highlights get carried over to all of the sizes, and mainly the take-home message from this is we can load a bioprocessing container or a BPC in the hardware within about 30 minutes. Even at 5,000 liter scale, we're able to use 2 technicians and load the BPC in about 30 to 45 minutes. We've been able to do that by making sure that the doors fully open, give you a lot of access to the inside of the reactor, as well as making sure that we have the holes within the reactors where the ports go and your sensors go very easy to see and make sure the tubing comes through those holes very well and robustly and uniform every time you load a BPC. Also, all these reactors can be integrated with our G3 Pro or G3 Pro series of controllers utilizing our TrueBio software, which runs on Delta V. And so that's one of the benefits that we bring with DynaDrive and our high-performance subs is the Delta V controlling aspect of the single-use bioreactors. Now, part of the design with the DynaDrive is different than other single-use bioreactors out there. It is a cuboid shape. We did that purposefully. One reason why is we get better fitment to the BPC within the hardware. The other thing is, and most importantly, is that the corners of the shape of the reactor actually act as baffles. So we get better mixing utilizing this shape. Also, the DynaDrive has a taller height to diameter ratio, which helps with your bubble residence time, and therefore it leads to better mass transfer, better KLA performance within the reactor. You'll see pictured there in the middle on this slide as well is an example of the design of the impellers for the reactor. You can see here we have 3 impellers that engage the cell culture fluid at different volumes, but we're able to get very good mixing at low RPMs, relatively low shear on your cells, and be able to mix well within the complete reactor system. As pictured there in the CFD, this is an example of our 50-liter CFD analysis that we've done, and we've now completed CFD analysis for our larger scales as well. So the design of the DynaDrive was really around performance, and the shape of the reactor helps with that to increase our performance for our power input per volume and our mass transfer capability. Another key aspect of the design is the Sparger. We utilize the drill hole Sparger, which has laser precision drilled holes in a piece of film. The drill hole Sparger is something that's not new. We utilize that on our HyPerforma, but we made this standard within our DynaDrive. One reason why we made it standard is you get better performance, you get the right size bubbles for the different sizes of reactors, so that the hole size in the drill hole Sparger varies depending on the size of reactor that you're using. Therefore, we can make sure that we have not only good oxygen delivery, but also good CO2 stripping within the reactor. One reason why we like the drill hole Sparge compared to, say, a Fritz Sparger is we get linear performance, as pictured there on the graph on the right-hand side of the slide. You can see in the black line that we have linear performance when utilizing the drill hole Sparge, compared to a Fritz Sparger that plateaus over time, and you don't get as much benefit at higher flow rates, and therefore it's a little bit difficult to predict. It's difficult to scale off of when utilizing the Fritz Sparger. And Qingwei will be covering our scaling later on in this presentation. But all of our reactors within the DynaDrive line utilize the drill hole Sparge, and that's one reason why we see very good scalability and good performance. As mentioned before, one of the design features of the DynaDrive is a lower turndown ratio. Pictured here is an example of what that really means. We're comparing the DynaDrive, which is at the bottom of this slide, which has greater than a 10-to-1 turndown ratio. You can see that we're able to go straight from an incubator into a 50-liter, and then from the 50-liter into a 5,000-liter. That's what we did a lot in the studies that Qingwei will cover. When compared to, say, a 5-to-1 turndown, which is part of our HyPerforma line, or even a 2-to-1 turndown, which also was our legacy HyPerforma, you can see that the DynaDrive cuts out a lot of connection points, a lot of assemblies that take place, also cuts out a lot of your other BPCs that need to be used within the system. So there's a lot of benefits that come from this lower turndown, and users, early adopters of DynaDrive, have recognized that they're able to save a lot of time and money when utilizing the low turndown of the DynaDrive. I'll now turn the time over to Qingwei, and she'll take us through the rest of the presentation showing the data. Hopefully my slides kind of gave you an example of the benefits of DynaDrive, and now she'll cover the cell culture studies that we've done, including the scalability from the 50-liter up to the 5,000-liter. So Qingwei, take it away.
Okay, thank you, Kevin, for a nice introduction. I think you covered very well for the capability of the DynaDrive subs. So I'm going to, take over, just directly go to the data package we have generated for the scalability study and for the comparability to the different subs. So for the scalability study, we choose 3 different subs: 50 liter, 500 liter, and 5,000 liter DynaDrive subs. In this table, listed the maximum volumes for the Turndown ratio, KOA, and mixing time, and the maximum P/V ratios for these 3 different subs. So for this study, we collaborated with the Kevin's team at Logan, and then the St. Louis PD team, and then St. Louis manufacturer. So, so for this study, we collaborated with the Logan team on the 50 liter, 5,000 liter, and 500 liter, those runs. And we choose 4 different cell lines for this study: 2 CHO-S cells, and 1 CHO-DG44, and 1 CHO-M cells. This is for 3 cell lines. They have different characters. 2 are the media production, 1's low and 1's high production. And then 3 are produce MAB and 1's non-MAB. And then they have different oxygen demand. So for this 3 cell lines, we call it 4 cell case studies. The case study number 1, we use the XP CHO cell, and this listed the process parameters when we run the experiment at 50 liter, 500 liter, and 5,000 liter DynaDrive bioreactors. As you can see, the seeding density, temperature, pH, agitation, DO, and the feeding strategy all listed here. And then the next slides showing the outcome from this study. So here the left side graph shows the VCD and the viability, and the right side graph shows the production, which is the titer. So here you can see for this XP CHO-S cell line, they had a pretty similar growth and production in the 50 liter, 500 liter, and 5,000 liter DynaDrive bioreactors. And the next slide shows the lactate and ammonia profiles for these 3 runs. And you can see the lactate profile, ammonia profile, and they are pretty similar and all within the historical range we have seen for this cell line. And you can tell from this chart, the 50 liter seems a little bit different from the 500 liter and a 5,000 liter sub. But all the data are within the historical data range we have observed. The next slide shows the PCO2 levels throughout the run, and then the online air and DO gas flow. So the 5,000 liter obviously, has slightly higher PCO2, which is, normal at the high, larger vessel. But you can see the level is quite well controlled throughout the run, or below 100 millimeter mercury for the PCO2 level. And the right side, the gas flow showing the VVM for both air and oxygen. The next slide shows the case study number 2. We use the Freedom CHO-S cells. This is a low produced cells. Showing the seeding density, inoculation volumes, temperature, pH, DO, and the feeding strategy, as previous case study showed. So similar format we used here. And this is the VCD viability titer. As you can see, 500 liter, 5,000 liter, they are closer and they had higher produce than the 50 liter. But this perhaps, 50 liter, because this is the first time ever we use the DynaDrive bioreactor to run any of the process. So sometimes the control wasn't, when we run the experiment, we did not control it as smooth as, when we had some experience. This slide is showing the lactate and ammonia profiles. So you see they are very well entrained for 3 different runs. The case study number 3, we used the CHO-DG44 cell lines. So the starting volume, seeding density, temperature, pH, DO, agitation, feeding strategy all showed here. Some parameters are scale independent, so they are using the same. So next slide shows the VCD viability and the titer. You can see again, they are very comparable from the different scales. And I want to point out that the 5,000 liter run for this cell line, we did terminate it early at day 11 because of some other issues, we identified during the run. But again, you can see the production, 5,000 liter produced quite well and is very similar to the 500 liter runs. Again, the ammonia lactate profile, they are very well entrained in the 3 different runs. And the PCO2 are also well controlled. It's well below the process limit. And then the air and the oxygen flow, VVM also showing here for all, these 3 runs, they are below 0.02 VVM for the oxygen demand. The case study number 4, we use the CHO-M cells, and in this slide shows the inoculation volume, seeding density, pH, agitation, DO, and feeding strategy. So the results showing in the next few slides. So here shows the VCD viability and the production. So they are very well aligned for all 3 runs. And then the lactate ammonia are entrained. The gas, the PCO2 are well below the process limit. And then the air and oxygen gas flow showing here. So for this process, we actually scaled up based on the VVM, and you can see the air and then the oxygen demand, and they are very well below the 0.025 for the gas delivery. So for this 3 different scale DynaDrive runs, we concluded, all these 4 cell cultures, they scaled up very well from the 50 liter DynaDrive to 5,000 liter DynaDrive bioreactors using the simple scale up strategy. So we use the constant P over V, and then the gas, we use VVM for the scale up. Similar VCD viability titer were observed for all, this different cell lines and from the different bioreactors. Metabolites and the gas, PCO2, we were kept in control in all different subs. The high turndown allow us to, run those runs to use the DynaDrive sub as N-1 and N vessel. So that's, as Kevin mentioned, this will save us the manipulation on different, connections, and then save the BPCs as well. So the next portion of the data package is going to talk about the comparison of different subs, which here we listed pilot scale subs. We used at XDR 50, XDR 200, HyPerforma 250, and DynaDrive 50. And we also look at the manufacturing scale subs, which is DynaDrive 5000, DynaDrive 500, HyPerforma 2000, XDR 2000. So this is a collaboration between St. Louis PD, St. Louis manufacturer, and Logan, Utah, Kevin's team for the DynaDrive development team. As Kevin mentioned early on, the DynaDrive used the cuboid off-center mixing strategy, and then the HyPerforma used the center cylindrical mixing strategy, and then the XDR used the off-center angled mixing strategy for those, both pilot scale and manufacturer scale vessels. So they all have different type of the mixing. For the performance, sometimes you will see a little bit of differences. But so here we wanted to show the process which we developed in the the process we developed for either XDR or for HyPerforma, but we transferred to DynaDrive. And, without doing a lot of manipulation, we just use a simple scale-up strategy and then directly transfer to these DynaDrive vessels. We wanted to see whether we can get success. Obviously, there's lots of optimization we can do in the DynaDrive vessels, but that's the next stage of the study. This is the stage we wanted to, transfer the process to DynaDrive vessels. So here listed the bioreactor capabilities for the pilot scale vessels: 50-liter DynaDrive, 50-liter HyPerforma, 50-liter XDR, 200-liter XDR, and 250 HyPerforma subs. We listed the maximum volume, turndown ratio, KOL mixing time, and then the maximum P/V ratios for all those vessels. And then these are the manufacturing scale bioreactor parameters. So 500-liter, 500-liter DynaDrive, 2,000-liter HyPerforma, 2,000-liter XDR, and a 5,000-liter DynaDrive. Again, we evaluated those with these 4 cell lines: XP CHO S, Freedom Choice, and the CHO DG44, and the CHO M cell lines. So these are the same 4 cell lines we used for the scalability study. And here we used this 4 cell lines at the different subs and look at the comparison. So the case study number 1, we used the XP CHO S cell line. And here listed the vessels used for this study, which is a 50-liter DynaDrive, 200-liter XDR, 250-liter HyPerforma, 500-liter DynaDrive, and a 5,000-liter DynaDrive. The process parameters are listed here: seeding density, temperature, pH, and agitation, DO, and then the feeding strategy. The case study number 1 here is showing the VCD viability. So you can see for this XP CHO cells, they grow very similarly in different type of vessels. And then the production are also very similar. This slide shows the metabolites, the lactate and ammonia. The 50-liter DynaDrive seemed a little bit off compared to other runs, but still within the historical data we have seen. And then the ammonia are very well in trend for all the runs. So we also look at the product quality comparison. So we use the capillary isoelectrophoresis to look at the acidic species, main species, and basic species of the product. And then we use SEC to look at the monomer aggregates and fragments to, compare this product quality from the different bioreactors. And you can see the monomer, they all above 94% of the monomer species, which is way above the acceptance criteria. And then the similar acidic, main, and basic species were observed from all different rounds. Case study number 2, we used the Freedom Cho S cell lines. And the vessels we used are 50-liter DynaDrive, 50-liter HyPerforma, 250-liter XDR, 2,000-liter HyPerforma, and a 5,000-liter DynaDrive subs. So the seeding density, temperature, pH, agitation, DO, feeding strategy, those parameters are listed in this table. The VCD viability and the production. Again, you can see the VCD viability, they are pretty well aligned from the different runs. And the titers is a little bit different on the production, but you can see the 5,000-liter DynaDrive and the 200-liter XDR, they produce the highest, while others slightly lower than this. So the lactate profile, ammonia profile are very well entrained from different runs. The product quality, again, we wanted to look at from all the different runs, but unfortunately, some runs, the final day sample were not collected for the product quality test. So we only were able to test it out to 200-liter XDR, 5,000-liter DynaDrive runs, those sample for the PQ analysis. So again, we use the capillary isoelectrophoresis and the size exclusion chromatography to look at the product quality. Both, the monomer percentage and the charge species, you can see they are very well aligned from 2 different runs. Case study number 3, again, we used the CHO DG44 cell lines. So this cell line, we have the most vessel type and most different, and volumes are used for this study. So you can see 50-liter DynaDrive, 50-liter XDR, 200-liter XDR, 250-liter HyPerforma, 500-liter DynaDrive, 2,000-liter XDR, and a 5,000-liter DynaDrive. All these vessels were used for this study. And the parameters, again, the process parameters are listed in this table. The VCD viability and the titer are showing in this slides. So most vessel size and the type, this is the most, the data-rich study here. And the similar viability were observed. The VCD, do have the highest VCD was seen in the 5,000-liter DynaDrive sub. The lowest were observed in the 200-liter XDR. However, the production, the 2,000-liter XDR had the highest production, and the DynaDrive were among the middle of the production range. But in trend, the larger vessel seemed to have higher titer than the smaller vessel. And then the lactate and ammonia profile are showing in this slides. So the fluctuation we observed for this cell line, for this runs. The product quality were evaluated again with the CIEF, SCC, and we also did the reduced CGE and the non-reduced CGE to look at the purity and the peak for this molecule. So you can see the charge variance seem that they have more consistent for larger vessels than the smaller vessels. And then the aggregation and fragmentation monomer, those were more consistent among all the vessels than other, PQs. The reduced CGEs seemed to have more variations than the non-reduced CGE, looking at the product quality. So, case study number 4. We use the CHO-M cell lines, and the vessels we used are 50-liter DynaDrive, 200-liter XDR, 250-liter HyPerforma, 500-liter DynaDrive, 5,000-liter DynaDrive. The VCD viability and the titer were shown in these slides, and the similar viability were observed, similar production were observed. In the VCD profile, we do see some differences, and then the XDR 50, both XDR 50 and 200 are the lower VCD vessels. They are the other vessel. So HyPerforma DynaDrive had a higher VCD than the XDR vessels. However, their production were quite similar. The DynaDrive 5000 actually had the highest production, 7.9 grams per liter for, this run. The lactate and ammonia profiles are showing here. So we do observe 2 different types of the lactate profiles, which, throughout the development we have seen this type of the lactate trend, but there's no impact on the growth. So we just left as what they are. And typically, the XDR had similar profiles than the other type of the vessels. The product quality were, evaluated using CIEF, SCC, reduced and non-reduced CGE. So for this CHO-M cell product, you can see the charge variants are very similar. All the samples have the greater than 92% of the monomer species using the SCC. The purity from the reduced and non-reduced CGE are very comparable. So, conclusions from this comparison study, we know this, 4 different processes which we developed for XDR and HyPerforma vessels. We successfully transferred to the DynaDrive subs without doing, mini manipulation. Process performance and product quality from the DynaDrive subs are very comparable from those, we obtained from the XDR and HyPerforma subs. So CHO-S, 2 CHO-S and the CHO-M cell lines showed enough robustness, which, allow us to transfer the process directly without much manipulation. However, the CHO-DG44, we do see some, differences from the vessel type to the growth, metabolites, in the production, or even product quality. So understanding the process definitely will help us, to have a better transfer. But for these purposes, we wanted to see, direct transfer from the, XDR or HyPerforma to DynaDrive, whether we can be successful. So that's what this study is for. And we did see, the success story on all 4 cases. So that's all the slides deck I wanted to present here today. There are a few, application notes we have published for these studies, and if you're interested, you can follow up with those links to check out and look at those details. So I'm happy to take any questions.
Anyway, Kevin, thank you so much. Great presentation. We're going to go to the Q&A now. So I would encourage anyone who is listening now live, if you would like to submit any questions, please do so. If we don't have a chance to get to yours live, we will follow up with you afterwards. So let's just get right into it here. Chingwei, I think this one is for you. What was the scale-up strategy for these runs?
Okay, yeah, so these are the proof of concept runs. So those are the very early, testing we tried to do on the DynaDrives. So for this, in mind, we wanted to use the simplest scale-up strategy as possible. So for this study, we did use the P over V, constant P over V for the agitation scale-up, and we use the VVM for the gassing strategy.
Okay, thank you. Let's see, we will go to, I think this one's for you, Kevin. I see that there's been a lot of work with CHO cells. What other cell lines have been used in the DynaDrive and at what scale?
Oh yeah, good question. Yeah, so we have various customers within the gene therapy realm using the DynaDrive, doing a lot of work with HEC293 cells. So far, I think the largest scale with those cells are up to 500 liter, with some plans of going up to the 3,000 liter and 5,000 liter. I just don't think those cell cultures have been completed yet. We've also done work with HEC293 in our facility here in Logan, Utah as well. And we have some application notes on the cell line. Also, we've done some work with SF9 insect cells. We have an application note using those cells as well for those interested.
Wow, really interesting. Thank you. We'll go back to Chingwei here. You mentioned that DynaDrive Sub allows to run at low volume. How do you handle the head sweep at low volume?
Yeah, that's a great question. So, when you run the low volume, the head space is huge. So to avoid that huge blanket of the CO2 or other gas, on the head, in those huge head space, you wanted to minimize that, if CO2 is the issue or, other gas is the issue. So for that purposes, to avoid that, we have considered the cross flow, when we designed the DynaDrive bag. So there's a cross flow Sparger design in the middle of the bag. So when we run the low volume, we use the cross flow Sparger as a head sweep. Therefore, your gas flow head sweep is at right, distance to your culture level. So this way we will avoid any of the gas buildup.
Great, okay.
So when we switch back to the high volume, then we can switch the head sweep to the normal head sweep. So that's how we, able to run the low volume and high volume in the DynaDrive. So we can use the vessel, for the low volume usage and the high volume usage. We use the same vessel.
That's really convenient. All right, Kevin, what studies have been done with higher cell densities? And then a follow-up to that is, how does the DynaDrive compare to the HyPerforma and XDR subs with high cell densities?
Oh yeah, another good question. Thank you. So the DynaDrive was purposely built to be able to support high mass transfer requirements and higher power input per volume requirements for higher cell densities. And so far we've seen densities well above 200 million cells per milliliter when operating in either ATF or TFF perfusion applications. We've seen that at the 50-liter scale. We've run a 500-liter perfusion culture for 60 days with an average about 150 million cells per milliliter in that. We've even had one user, 50-liter DynaDrive, get above 300 million cells per milliliter, and they even had more oxygen capability within the system. So users that are taking advantage of this higher capability reactor are seeing the benefits of that with cultures well above 100, 150, and even 200 million cells per milliliter. So far, the largest reactor that we know that's gone to higher cell densities is that 500-liter run that we've done here in Utah. We based on other analysis we've done, we were confident that the 3,000-liter and 5,000-liter systems could do higher cell densities as well. So we just haven't done those cell cultures, but we have the data for the 50 and the 500. You know, how it compares to our legacy HyPerforma sub, there is more capability within the DynaDrive to support higher cell densities. The higher column height leads to better bubble residence time, therefore the KLAs are higher. As I mentioned, we have about double the KLA, double the power input per volume, so that you can support the higher cell densities, even up to the 5,000-liter scale. The HyPerforma, our legacy HyPerforma system, does really well with higher cell densities up to about the 500-liter scale, where we introduced a perfusion enhanced model of our HyPerforma sub a few years ago, up to 500 liters. So we can get around 150 million cells, maybe pushing 200 million, up to 500-liter volume with our HyPerforma, but anything higher volume than that or higher cell densities would require the DynaDrive, and that's one reason why we designed the DynaDrive the way it is.
Great, thanks. I will leave this up to either one of you. What is the best way to overcome challenges when adopting new technologies?
I can take on this one. So, I mean, most people probably are hesitant to, take the new technologies just without, confidence. But hopefully this data package will convince you that DynaDrive with such larger capability on the mixing and the gas delivery strategy, will convince you that DynaDrive is easy to adopt and easy to allow you to transfer your existing process to the DynaDrive bioreactor without, manipulation. Obviously, DynaDrive has a much larger capability than other single-use bioreactors, and you have a lot more room for the optimization. Even some process we had a challenge with, existing subs, we are allowed, there's a lot of room for improvement if you do the DynaDrive. But I mean, the overall, the take-home message is DynaDrive is easy to adopt. You can directly transfer your existing process to DynaDrive, and also DynaDrive will allow you to do the further optimization.
Okay, great. So I don't mean this one to be a trick question for you. What opportunities does new sub technology provide? And I know, Kevin, you've already mentioned, the higher cell densities. Chinwei talked just in the last one about larger volumes. Is there more than that beyond?
Well, I'll just answer that. I think I mentioned the higher cell densities. That's a key one. It allows maybe users in the PD space that have some processes that they're able to have, high cell densities at small bench scale reactors, whereas previously they couldn't scale those processes to larger volumes because single-use bioreactors didn't have the mass transfer to support those volumes. Now with newer technology, it allows for those processes to be scaled up in a manner that that the reactor is capable of supporting those processes without risk of running the reactor, say, at the very limit. So I think that's one of the opportunities, is it allows for new processes that maybe had previously been able to be done at a bench scale but hadn't been able to be scaled up, now are able to be scaled up to the larger volumes.
Okay, so follow up to that, how does it affect shear? Oh, no problem. So the new sub-technology, speak in reference to, I know it has a more efficient mixing. Does that lead to less shear then?
Yeah, it does, because of the way that it's designed with the multiple impellers and the corners that act as baffles. We're able to run it at lower RPMs, and therefore the tip speeds of each impeller is less than you might see with a reactor that has a single impeller, and that helps reduce shear. Then the other thing is around the sparging technology. There's a lot of shear that occurs typically with micro Fritz spargers as those bubbles break, especially if you're trying to run those at high gas entrance velocities. That's where a lot of shear can happen on the cells. And so what we found with the drill hole sparger that we use in both the HyPerforma and the DynaDrive, we can run at lower gas entrance velocities, and therefore reducing the shear that's caused by the sparger. And then one efficiency that we get with the DynaDrive compared to, say, our legacy HyPerforma reactors is because we're able to maintain the bubble residence time for longer, we do not have to sparge at the same rates that we had to in our legacy reactors, and therefore we can have a lower rate of that gas entrance velocity, also therefore reducing shear within the reactor itself. So we've looked at both tip speed and that gas entrance velocity through the spargers as being key components to shear, and we've reduced those quite a bit.
Great. Okay, so I have another question here. Does DynaDrive Sub have anti-foam sensors so that it can avoid high foaming during the cell expansion process? And can the system be set to feed the anti-foam automatically?
Yeah, great question. It does. Yeah, each one of our BPCs have a sensor in it that detects foam, and that can be controlled with the controller with an anti-foam pump. And therefore, when the sensor is alarmed, anti-foam can be kicked on and introduced to the culture. So we actually have an application note using that foam sensor in our legacy HyPerforma line, but the same thing applies with the DynaDrive, where we've actually found that by controlling anti-foam introduction in this manner reduces the amount of anti-foam used significantly, which is helpful for your downstream people. And so we've carried that technology over to the DynaDrive BPCs as well.
Okay. I have a question here from Matthew. How custom configurable is the DynaDrive BPC?
Yeah, good question. As far as the line sets are concerned, we can customize those pretty much however you want. We are trying to keep the chamber as standard as possible, but when I say standard, I have multiple standards of each size available. So so far we've been able to keep the designs that users have come up with within our chambers, but then we're customizing the line sets to fit your needs. And so kind of look at it as a case-by-case basis and try to stay within standards where we can, because that will ultimately help reduce lead times and costs over time, because we can end up stocking those chambers. And that's our plan, but like I said, I have multiple standards of each size that accommodate most needs that we've found so far.
All right, great. Looking through what else was submitted, we have some very specific ones. So I think, Matthew, who asked that question, has a follow-up, but I think we're going to contact you directly on that. Want to mention to everyone that there are links to a 3-part series on scale-up evaluation. It's available in the related columns window, which is directly below the media player. Want to thank everyone for attending. As I stated before, if we didn't get to your question or it's very specific or requires more explanation, we will definitely contact you privately after the webinar. This webinar will also be available on demand in about 24 hours. You'll receive an email when that is available. To everyone, to our presenters, thank you so much. Great presentation, lots of good info there. Thank you all for joining us, and we hope to see you again on another webinar. Hope you have a great rest of your day.